Hi students, welcome back to yet another lesson in biochemistry. Today we will be discussing about lipoprotein metabolism, which is a subpart of a lipid metabolism, and it is covered under biochemistry competency number BI 4.4. If you go by the National Medical Commission, that is NMC curriculum for first year, it is under 4.4. We will be covering the whole lipoprotein metabolism under 2 classes, so it will be easier for you to grab the concept and process what we tell in this class to continue with the next class. All right, so to start with, we'll ask ourselves, what is the problem? The problem being, what are lipids? Okay, this is a very basic question that are asked in Viva. So lipids are we know something which is not soluble in water. There is a whole classification, that is lipophilic and hydrophilic, right? So if the lipid is not loving water, then the question is, how does the lipid molecule get transported? Via aqueous medium, because our blood is water, right? The answer to this problem lies in lipoproteins. Okay, because the lipid are actually arranged in such a way that they form a friendship, they join hands with part of a protein. How? The various apoproteins cover the core, the core which is formed by lipophilic substance, and they are coated by phospholipid and apolipoprotein, and thus they become hydrophilic. The entire setup becomes hydrophilic. Okay, so this is the basic structure of a lipoprotein. If you are not concerned about lipoprotein, most of you already know what is a lipoprotein, but for those to whom this term is new, lipoproteins are compounds or molecules whose core is made up of triacylglycerol or TG or triglyceride, same thing, and cholesterol ester. The surface is made up of phospholipid and some cholesterol. Apoproteins, those are the main regulators of lipoprotein metabolism that we will be discussing very soon. And what are the types of lipoproteins? Chylomicron, VLDL, that is very low density lipoprotein, IDL, intermediate density lipoprotein, LDL, low density lipoprotein, and HDL, that is high density lipoprotein. So we'll be discussing them one by one. I'll be showing you what exactly you need to know, right? Now, for MCQ purpose, what you need to know, the example of lipoproteins that are rich in triglyceride. These 3 are the bonds: very low density lipoprotein, VLDL, that is VLDL remnants, okay, and intermediate density lipoprotein. They are very much rich in TG, right? And the small or dense LDL or the normal LDL, those are not rich in triglyceride, they are rich in cholesterol, right? So how do we can measure them? With the help of various apolipoprotein. We'll be seeing very soon what type of apolipoproteins are present in what lipoprotein. Okay, this is the chart that you need to memorize. Okay, there is no secret in how you can memorize, but I'll tell you a few important areas which are must know, because remembering the entire thing is actually impossible. It may be possible if you study only this, but we need to study a lot more before appearing the first prof exam, right? So you should remember that B48, that is apolipoprotein, or we in the short form we say apo B48 is present in chylomicrons only, and B100 is common to all these 3, VLDL, IDL and LDL. They are commonly known as B100 containing apolipoproteins, or B100 containing lipoproteins, because B100 is itself an apolipoprotein. HDL does not contain apolipoprotein B. Any form of B, whether it is B48 or B100, it is not present in HDL. So what is present in HDL? In HDL, ApoA, ApoC and ApoE, these are present, right? And I already told you in the previous slide, the first 3 are the triglyceride rich lipoproteins. So the main lipids in them are TG, okay? And for the last 3, The main lipids are cholesterol. IDL, since it is intermediate, it contains both triacylglycerol and cholesterol, right? One more thing to know, this, what is the source, what are the various sources of lipoprotein? If you look into this chart, chylomicrons are formed in the intestine, VLDL is formed in liver, IDL is formed from VLDL, LDL is also formed from VLDL, and when it comes to HDL, it is again formed in the liver, intestine, chylomicron and VLDL. There are multiple sources from which HDL, the components of HDL are donated and HDL is produced. You should also notice one thing, that as we go down, the size decreases. The diameter for chylomicron is maximum 92,000 nanometer, and it is least in case of high density lipoprotein. So the size decreases and therefore the density increases. So chylomicrons have got least density, and HDL, as the name suggests, high density, has got the highest density, right? Next, if you look into the composition of protein, you should always remember the most and the least. The one that contains most amount of protein is HDL, and the lipoprotein that consists the least amount of protein are chylomicron. So chylomicrons contain more lipid, see, 98 to 99%. The lipid portion is much more in chylomicron compared to the protein part, whereas in case of HDL, the lipid portion is the least and protein portion is much more. You know, LDL is known as bad cholesterol and HDL is known as good cholesterol. Okay, I hope you know this. We'll be explaining it very soon. Anyway, so another thing that is asked in Viva, that lipid metabolism occur in three major areas. What are the areas or the tissues where lipid metabolism mainly occur, or more specifically lipoprotein metabolism? So the areas, the tissues that are involved are intestine, liver and the extrahepatic tissues, that is muscle and the adipocytes or fat cells. So any other organ or any other tissue that is given to you as an option in the MCQ will be ruled out. Next, we'll look briefly how a chylomicron is synthesized, it is produced, and then we'll look into the detailed cyclical metabolism, how it is happening. All right. So when we eat dietary fat, they break down with the help of various lipases. All right. So gastric lipase, intestinal lipase. Those lipase means an enzyme that is a hydrolase, that with the help of water that breaks a lipid into its simpler components, that is fatty acids and glycerol. So 2-MAG means monoacyl glycerol, where the acyl group is present in the second carbon. So fatty acids and 2-monoacyl glycerol excuse me, they are absorbed from the brush bordered cells of the intestinal epithelium. These are the intestinal luminal cells. So they get in, and with the help of ATP they get converted to TG or triacylglycerol, and to that TG apolipoproteins are added. What are the apolipoproteins? B48, C and E. Okay, and thus chylomicrons are produced. Okay, so again, if you look into the structure of chylomicron, it is a huge structure, very less dense, a huge amount of lipid in the core and a very small thin coating of protein, right? And the chylomicrons are absorbed from the basolateral side into the lymphatic system. Okay, now before going into the chylomicron assembly itself, we should know about what are the various forms of apolipoprotein B. I already told you during discussing the chart, there are two forms of apolipoprotein B. One is 48 and one is 100. What is this 48 and 100? These are the number of amino acid chain that is attached in the protein. And this is basically the same protein, the same parent, one in which 100 amino acids are added sequentially during translation, and in one, after 48 amino acid addition, the translation stops. This control is happening via RNA editing. Okay, it's the post-translational modification or even post-transcriptional modification to some extent, and we will be discussing them in detail in our molecular biology classes So for now you should know two forms exist. The B48, the smaller one, is attached to chylomicron because chylomicrons are produced in the intestine, and the B100 is attached to VLDL and all other lipoproteins that are produced in the liver. Okay, so when to start with, chylomicrons are attached to apolipoprotein B48, and gradually in their course they are picking up ApoA, ApoC and ApoE while circulating in the plasma. Okay, and the triglyceride composition of chylomicron, it very closely resembles what we eat in our food. So if we take a huge fatty food, the amount of chylomicrons that will be produced are a lot of chylomicrons will be found in the plasma. And if we avoid fatty food, the amount of chylomicron immediately if tested in blood after food will be much less. Now let us look into the cyclical assembly of how chylomicrons are produced and what are their metabolic fate. This will come as a long question. Okay, metabolic fate of chylomicron will come as a long question, and you should give a schematic diagram for that. Diagram first, then you need to write. Even if you do not have time, if you give diagram, you will get much more than pass mark. Examiner will be very impressed because the pictorial based answers gather much attention, and it proves that you have a clear concept. Even if you know the whole thing, even if you write 1-page note, the student who has given a good diagram will get much more than a just descriptive essay type answer script, right? So to start with, dietary triglyceride, I already shown you the brush bordered cell. The chylomicrons are formed and they get absorbed into the lymphatics. Over there it is known as nascent chylomicron. It has got a core of triglyceride and cholesterol, and the apolipoproteins that are attached are ApoB48 and ApoA. Alright, so what happens to it? In the next step, this nascent chylomicron collects ApoC and ApoE from HDL. This is a common formula which we will apply. Mind it, HDL, it donates ApoC and ApoE. You can consider HDL or high density lipoprotein as a universal donor of ApoC and ApoE. Okay. So in that process, what happens? The nascent chylomicron is converted to mature chylomicron. So now it is having B48, it's having ApoE, it's having ApoA, it's having ApoC, right? Now this chylomicron reaches extra hepatic tissues and is acted upon by lipoprotein lipase, which breaks down the triglyceride content which is inside the core. Okay, you can see the volume, the amount of circle has reduced, because why? The amount of triglyceride is reduced as they are digested or they are acted upon by lipoprotein lipase. What this lipase do? They convert the TG into fatty acids and glycerol. This fatty acids and glycerol have got their own metabolic fate. Of course, fatty acids undergo beta oxidation, glycerol is acted upon by glycerol kinase and they can enter the TCA cycle. They can be converted to dihydroxyacetone phosphate. That's a whole another metabolic pathway that might be asked in exam anyway, because everything is interconnected. What we need to know is this, after action of lipoprotein lipase, it gives back ApoA and ApoC to HDL. So HDL was donating C and E, HDL is receiving A and C, right? What the chylomicron becomes? So after donating A and C, the chylomicron is left with ApoB48 and ApoE. Mind it, this two looks similar, right? But the size of nascent chylomicron is much bigger. It has got ApoA with 48, whereas the chylomicron, now after the action of lipoprotein lipase, the chylomicron has now got B48, but it has got ApoE, and the size is much smaller and it becomes much denser. It is known as chylomicron remnant. Okay, so what happens to it? the this chylomicron remnant is now actually absorbed by the liver. It finally is delivered to the liver. How? Because there are multiple receptors in the liver that can identify these various apolipoproteins as ligand. Ligand means something which is attached to a molecule which can be identified by a specific receptor. So over here, the receptors in the liver, that is in the hepatocytes, they accept or they recognize B48 as a ligand, they recognize ApoE as a ligand, and thus they can absorb the whole chylomicron remnant. You can see LDL receptor, they are recognizing LDL receptor, they do have recognition for ApoE or ApoB100. So in this case it is engulfing the chylomicron remnant by recognizing ApoE, right? Also there is another protein that is known as LRP or LDL receptor protein. So both of them are actually helping in absorption of this chylomicron remnant. Going inside, it is acted upon by hepatic lipase. This HL is known as hepatic lipase. This hepatic lipase now is much stronger in action, and it also degrades the whole chylomicron remnant, and it converts the whole thing to cholesterol and fatty acid, because it is breaking down the TG into glycerol and fatty acid, and also the cholesterol that is in the core is released into the liver. Alright, so this actually completes the chylomicron metabolism or metabolic fate of chylomicron. So this is a long question. This diagram has been taken from Harper's illustrated textbook of biochemistry, and believe me, if you want to study lipoprotein metabolism, Harper is the best book in my opinion. Of course, any textbook is best provided you remember the whole thing and it has got all the points covered. Many textbooks omit many essential points for undergraduate. I mention you, there are multiple sub things that are happening, but you don't need to know all, but you cannot omit essential things as well, right? So these are the metabolic fate of chylomicron. What will happen with the ApoE? This ApoE is actually again donated to HDL because HDL is formed from liver. So it's the HDL is formed from liver, and then it can donate C and E to the nascent chylomicron, right? So this is chylomicron metabolism. I want you to pause this video. You can take a screenshot. You can mentally map the whole process, and when you are ready, we'll go forward with the next step. The next step is the metabolic fate of VLDL, and it catches up right where chylomicron metabolism is completed, because this is a whole continual thing. One thing gets converted to another, and at the end the less dense and bigger sized chylomicron is converted into more dense and small in size HDL. Okay, so excuse me. So to start with, VLDL, from where it is produced, if you remember the chart, it is produced from liver. Okay, so at first the particle that is formed from the liver is known as nascent VLDL. So nascent VLDL is again a lipoprotein, definitely, that has got triglyceride and cholesterol at its core. It has got ApoC and ApoE along with apolipoprotein B100. So a lipoprotein which has got B100, ApoE and ApoC as well as TG and cholesterol in its core is known as nascent VLDL. Now if you want to relate to chylomicron metabolism, it is almost similar, because just the difference is chylomicron was getting synthesized from intestine, here it is getting synthesized from liver, but the next step is absolutely similar, that is ApoC and ApoE are being donated from HDL. Yes, it already had ApoE, but the amount of ApoE and ApoC are getting enhanced. See, now this nascent VLDL becomes mature VLDL, alright, or it is very low density lipoprotein. The final form of VLDL is this. If you want to compare the two, this step is absolutely similar. HDL is donating ApoC and ApoE, right? So what was the next step in chylomicron? If you want to relate to both of them side by side, it is very easy to remember if you can remember one. it is reaching the extrahepatic tissue where it is acted upon by lipoprotein lipase, which is degrading this some part of the triglyceride, and it is converted to fatty acid and glycerol, right? And after that, this is where it gets different from the chylomicron. So over here, ApoC is again getting, it is again received by HDL. Okay. Next, this is converted to another form of lipoprotein, which has got, having lost the ApoC, it is now left with B100 and ApoE. This is known as IDL or intermediate density lipoprotein or VLDL remnant. It is the same thing. Okay, the structure is very similar, but what has changed from VLDL to IDL? Number one, ApoC is gone, and number two, the amount of triglyceride and cholesterol, actually the amount of cholesterol increases, but the density, the size becomes small, hence the density increases. Okay, so this IDL or VLDL remnant can be directly engulfed or directly be incorporated into the liver hepatocyte. Why? Because this LDL receptor can identify this ApoB as ligand. So whenever a lipoprotein has got B100, it can directly go into the liver. Or what can happen with that? There is the variant of the metabolic fate. This can be converted to LDL. How it is converted to LDL? By just giving away the ApoE. Who can accept the ApoE? Of course HDL, because HDL can accept ApoE. This ApoE can also be accepted by hepatocytes, liver. So what is the change that is happening from IDL to LDL? Note, the TG is gone. Okay, so LDL, when it comes to low density lipoprotein, the core that is made up of TG and cholesterol in IDL, it's completely gone. Only the cholesterol is remaining, along with only one lipoprotein, that is ApoB100. So remember, I told you initially that TG rich lipoproteins are chylomicron, VLDL and IDL, or chylomicron, VLDL and VLDL remnant, and rest, that is IDL, LDL and HDL, are having only cholesterol. Okay, IDL, since it is intermediate, it has got both TG and cholesterol. So what are the metabolic fate of LDL? Again, LDL, since it has got ApoB100, okay, it is identified by number one, liver, because LDL receptors can identify ApoB100 as a ligand. It can directly go into the liver, or this LDL can or is actually deposited in the extrahepatic peripheral tissue, that is the arteries and other various intimal wall of artery and other nerves. That is why it is known as bad cholesterol, because excess deposition of LDL cholesterol will lead to arterial blocking, that will cause cardiovascular accident. It can be deposited on the skin. You see people with a very high amount of LDL cholesterol will have white plaques around the eye, that is known as xanthelasma. Okay, xanthema and xanthelasma. So all those are possible once LDL is formed and it is deposited into the extrahepatic tissues. Okay. And in liver, what happens? The final destruction in liver and extrahepatic tissue, these are the various target organs, that is lymphocyte, fibroblast and endocyte. They actually help in final destruction of the cholesterol. How? They are oxidized to form foam cells. That is all whole another dimension. We are not going into that, but just remember, LDL can be deposited in the peripheral tissue, or it can directly go inside the liver, either LDL or IDL. And over there, the hepatic lipase, again hepatic lipase you can label over here, give a small square, you can write HL, just like the figure of chylomicron, and those are helping in degradation of the TG into fatty acid and cholesterol, or in case of LDL, the cholesterol simply gets detached. Now this cholesterol, if it is coming to extrahepatic tissue, it is deposited in the arterial wall, but in liver it is used in the formation of bile acids. Alright, so not only LDL is depositing cholesterol in the extrahepatic tissue, it also plays a role in depositing cholesterol in the liver, which can lead to a condition known as fatty liver, because excess fat deposition in liver is also fat. Alright, so we can also visualize the same with the help of a very cute animation. So this animation actually shows how VLDL are metabolized and they enter the circulation from the liver. So number one, VLDL are produced from the liver. They are having ApoB 100 and they are actually getting ApoC and ApoE from HDL, specifically the ApoC2. Okay, if you want to score good marks, you need to mention ApoC2. So ApoC2 and ApoE are donated to the VLDL, and thus the VLDL remnant are becoming mature VLDL. Then they are acted upon by lipoprotein lipase in the tissues, which degrade the TG and cholesterol, and they are collected into the they go into the extra hepatic tissues, and having lost the triacylglycerol, they are converted to LDL. There is an intermediate step, that is IDL, intermediate density lipoprotein, which contains equally amount of TG and cholesterol. But majority, the ultimate fate is production of LDL after donating the ApoE and ApoC2 to the HDL. So the lipoprotein only remains as a cholesterol core and B100 as the apolipoprotein. So once LDL is formed, it has got two metabolic fate. It can be taken up by the liver or with the help of LDL receptor, or it can be used in delivery of the cholesterol to the extrahepatic tissue, both cholesterol and the triacylglycerol, right? So this now brings to the question, an explanatory type of question is often asked, why LPL or lipoprotein lipase is known as metabolic gatekeeper? Okay, this can be asked in Viva, it can come in theory. It's because the number one thing is the whole process of LDL synthesis from dietary fat starts from chylomicron, right? And if there is excess chylomicron in the body, then the triacylglycerol cannot be cleared, right? But lipoprotein lipase actually helps them by breaking down the triacylglycerol and converting them into fatty acid and glycerol. So if there is deficiency of lipoprotein lipase, it means what? In the first step, the chylomicrons cannot be acted upon at the extrahepatic tissue, and the whole amount of TG that is inside them continues to be circled and deposited in the liver. This condition is known as chylomicronemia, where even after small amount of meal, it may be non-fatty, there is a huge amount of chylomicron that is present in blood. This is a type of familiar hypercholesterolemia. So in lipoprotein deficiency, excess cholesterol will be produced because it cannot be cleared by lipoprotein lipase. So these are the few states by which lipoprotein lipase is regulated. You just need to remember, if you can remember one, the opposite will be automatically remembered. Just remember, in fed state, lipoprotein lipase synthesis increases, the synthesis and activity both increases in adipocytes, okay, and its activity decreases in skeletal muscle and heart. Remember, the way lipoprotein lipase acts, it's reciprocal in adipocytes and in skeletal heart muscle, because skeletal heart muscle are using fatty acid as a food, okay, and in case of adipocyte, they are actually using these fats to produce the hepatocytes, adipocytes, right? So when we have already eaten much, the fat cells can actually increase the store by synthesizing more and more adipocytes, and so it needs lipoprotein lipase. But in case of fed state, when you are well fed, the skeletal and heart muscle do not need fatty acid for energy. Hence, lipoprotein lipase level is actually low, right? The reverse will happen in fasted or in state of exercise. So in fasted and exercising state, in case of adipocyte, we don't need to synthesize the adipocytes, right? So decrease lipoprotein lipase activity. But in case of muscle, the lipoprotein lipase activity is increased because we need more and more fatty acid to give us energy with the help of beta oxidation, right? Because already in case of fasting state, the energy production from glucose is done, whereas in fed state there is enough glucose to produce supply energy to the heart and skeletal muscle. One exception is in case of lactating mammary gland. It is always in a state of hypermetabolism, that there is a huge increase in lipoprotein lipase activity. Okay. So one interesting thing that examiners, classical biochemists, I would like to say, have got a very soft corner for, that is Brown and Goldstein. They actually discovered this whole cholesterol receptor mediated pathway, the cholesterol homeostasis, and they got Nobel Prize in 1985. Incidentally, this is the year where I was also born, right? So we come to an end of the first class. We have covered in detail about how lipoproteins work, what are the various types of lipoprotein, what the comparison of various type of lipoprotein, the apolipoprotein components. We have told you what are the sources, how chylomicron is formed, what is the metabolic fate of chylomicron, what is the how it continues into VLDL metabolism, what is the metabolic fate of VLDL. If a question comes, metabolic fate of VLDL, IDL, LDL, the same diagram will cover all. Alright, and I also told you how LDL is a culprit in depositing cholesterol to the peripheral tissue as well as in the liver. And we also told you what are the regulatory fate of lipoprotein lipase, how it is regulated, and why it is known as metabolic gatekeeper. Or simply you can get a multiple choice type question, who is known as metabolic gatekeeper, and the answer will be lipoprotein lipase. And I also told you a fun fact, that is in 1985 scientists Brown and Goldstein got a Nobel Prize for discovering the pathway which we are studying. So I thank you all for your patient hearing. In the next class we will be discussing about CETP, cholesterol ester transport protein. We will be discussing about HDL metabolism, very important. We will be discussing about liver reverse cholesterol transport. We will be discussing good cholesterol, bad cholesterol, lipotropic factor. There are so many things to cover. It will be very easy provided you have already you are well read with today's information. I will see you soon with another video. Till then, bye and take care. Oh yes, I just forgot, we have now got a good Facebook group, the link to which is given in the description, where you can actually join and participate in discussion with other colleges, students of various colleges across India. The link is in the description, and we have got multiple playlists in my YouTube channel. So something which I have already covered, you can look into those playlists. There are multiple, every type of metabolism, molecular biology techniques, all have been covered in detail. So feel free to look into that. Please like the video if you have genuinely liked my content. I expect you all, if you are attending the next class, be well read with today's content. Anyways, again I'll say bye over here. See you soon and take care.